Thank you for attending Texas Biomed's first Global Health Symposium. I'm Ricardo Carrion Jr. I'm Professor and Director of Maximum Containment Contract Research at the Institute. The goal of this symposium is to bring together thought leaders in science, public policy, global public health, and philanthropy. It is through true collaboration with one another we can share, learn, and replicate new effective innovations and create greater collaborative networks. Texas Biomed's mission is to pioneer and share scientific breakthroughs that protect you, your family, and our global community from the threat of infectious disease. This symposium is an extension of that mission. By reflecting on advances, past, present, and future, we can learn from one another and build forward from this pandemic better and help educate our communities that health starts with science. It's important to know what scientists are seeing on the horizon, so I'm honored to host this panel of experts. Now, at least one or more of the scientists I'm about to introduce has been at the forefront of every single outbreak and every significant pathogen over the last 40 years. First, we have Miles Carroll. Miles is Director, Head of Research and Development at Public Health England. He joined PHE in 2008 with a joint position at the University of Oxford. He earned his PhD in HIV vaccine research from medical faculty at the University of Manchester. He then started a fellowship at the NIH. For the last decade, he has focused his work on emerging viral pathogens, real-time sequencing to support molecular epidemiology and outbreak response. In addition, he has led the development of in vivo models for high-consequence pathogens and for vaccine development. We also have joining us Dr. Erica Ollmann Sapphire. She is professor and president-elect at the La Jolla Institute for Immunology in La Jolla, California. Erica earned an undergraduate degree from Rice University and had a dual major in biochemistry and cell biology. She received her PhD from Scripps. Her focus is on how and why viruses like Ebola and Lassa are pathogenic and provides a roadmap for medical countermeasures. Erica has also led the Viral Hemorrhagic Fever Immunotherapeutic Consortium and is director of this organization. This consortium is an NIH-funded Center of Excellence in Translational Research and unites 44 competing academic, industrial, and government labs across 5 continents to understand and provide antibody therapeutics against Ebola, Marburg, Lassa, and other viruses. Recently, Erica was asked by the Bill and Melinda Gates Foundation to head up Coronavirus Immunotherapeutic Consortium and is working with Operation Warp Speed to advance antibody therapeutics against SARS coronavirus 2. And finally, rounding out our panel, we have Dr. Connie Schmaljohn. Connie is director at the NIAID Integrated Research Facility at Frederick. Prior to that time, she was senior research scientist for medical defenses against infectious disease at the U.S. Army and directed the research program at USAMR, that is the United States Army Medical Research Institute of Infectious Disease. Connie had earned her BS from Nebraska, a PhD from Colorado State, and she had joined USAMR initially as a National Research Council postdoctoral fellow. Connie has served as president of the International Society of Hantaviruses. She's chaired the American Society for Microbiology Biodefense Conferences. She's also chair of the National Committee on Taxonomy of Viruses. She serves a number of advisory roles and advisory boards, including Interagency Public Health Medical Countermeasures, Board of Scientific Counselors for the Vaccine Research Center, and a scientific advisor for CEPI, the Coalition of Emergency Preparedness Innovations. She was selected to the Academy of Microbiology in 2007, selected as a fellow of the International Society of Vaccines in 2015. She received Order of Military Merits in 2002. She received a President Rank Award in 2007. Thank you, panelists, for joining our discussion today. First, we will now have Miles Carroll perform the first presentation. And the title of his talk is Emerging High Consequence Viruses, Threats, Unknown Unknowns. Thank you, Miles.
Great. So thank you very much for the kind invitation to speak and the introduction there, Ricardo. And hello to everybody in Texas. I wish I was there personally. I've been to your institute before and had a fantastic time. I'm stuck in England, about 100 miles south of London, but at least the sun is shining and there's some warm beer waiting for me at home. So I've got 10 minutes and I'm going to try and give you a couple of key pointers on what we do know, and then obviously what we don't know, and what we've learned from the outbreaks to help us for the future. So this is a summary of high consequence virus outbreaks in the last couple of decades. It's got all the usual suspects there, from Ebola virus, Marburg, Crimean Congo hemorrhagic fever, your avian influenza, and then of course it's got SARS-1, MERS, CoV, and then the significant pandemic of today with SARS-CoV-2. But what I think I want to highlight is that this is probably 1/100th of what really happens. So we only see those outbreaks that cause significant disease, human death, that allows us then to see a hot signal, to go in, take samples, work with the local health authorities in those countries, do the molecular diagnostics, and hopefully sequence and identify these pathogens and put a label on a map. Most of the time, that doesn't happen. People die of infections all the time in isolated villages, deep in forests, etc., and we never hear about it. And another statistic is that it's estimated that we know of only about 0.1% of all those viruses that actually exist in nature. So we've got a huge amount to learn, and that's something I think we need to invest in the future. So there's a couple of general patterns of these high consequence emerging pathogens. And that is that they're usually RNA viruses. I know all the pox biologists will say, well, monkeypox and smallpox, they aren't the DNA viruses, but most of them are RNA. And that gives obviously about a 10-fold higher mutation rate because of the lack of proofreading systems for replication of those genomes. And that allows really good genetic adaptability, so the virus can change in a relatively low number of replications to a new host, if it can get into that new host in the first place. Another key property of these pathogens is that they are usually harbored in an animal reservoir, and they're zoonotic, and they spill over into humans. And it's okay, so I'll just avoid bats and other creatures, but sometimes you can't avoid them because a lot of them are vector-borne. With the help of ticks and mosquitoes, etc., you don't really have much choice if these pathogens are going to reach you at some point. So that's just the point that, well, obviously, there's a huge number of viruses out there. We know only a fraction of them, and a huge proportion of them have the potential to cause disease in animals as well as humans. I stole this lovely schematic from CDC, and because it does summarize so well these zoonotic and spillover events that occur continuously around the world. And for example, for Ebola virus, we didn't used to know for sure, but we feel a bit more confident now that Ebola virus does have a reservoir in certain species of bats. We also know that it's able to infect other forest animals as well. And via the bushmeat hunter, who is going locally to look for meat sources for his family and his village, inadvertently will pick up infected animals, take those to the village, and the virus will spread subsequently. And then with the survivor in the top right-hand corner there, we know that for this virus anyway, it doesn't just disappear after the infection's over. It can persist in certain immune privileged sites and may cause reinfection subsequently. the point of this slide is to highlight that you can get to good diagnostics to the site of an outbreak, but you need to be told the outbreak is in the first place. So this is the going back to December 2013 for the outbreak of the West Africa pandemic. It did reach each continent and would be theoretically classified as a pandemic. So it was 34 people were reported through ProMed to have died from a mysterious hemorrhagic fever before samples were shared with some European laboratories, and diagnostics were performed to show it was Ebola Zaire and not Lassa fever as thought at one point. But within a week of that positive diagnosis, we had, as members of the European Mobile Laboratory, which responds specifically to high containment pathogens in the African continent, we had a diagnostic lab there in the bottom right-hand corner doing pan-filovirus Automa PCR, which was obviously a great help to MSF in trying to differentiate who had malaria, who actually had the virus. But obviously, by this time, respectively, it was too late. The virus has already spilled over into neighboring countries of Liberia and Sierra Leone. What has really helped in outbreak investigations is the real-time epidemiology, so real-time sequencing that has enabled molecular epidemiology to be supported anywhere in the world. And we've used these mobile sequencing units from Oxford Nanopore technology, which many of you would probably be using today. But as you can see the date, this was back in May 2015, and we did the first Ebola sequencing in Guinea using one of these devices after getting the kit into the country within 14 hours. We set up a real-time sequencing capability with WHO and the local Guinean authorities, and this was used in anger in many cases to identify mysterious transmission and then shut down those chains of transmission, including coming over from borders for Sierra Leone and Liberia. I think one of the best applications of real-time sequencing for outbreaks was back in March 2016. This is best part of a year when the forested region of Guinea was supposed to be clear of Ebola. A young lady was found dead in the community. A very smart healthcare worker took a swab. That swab was then sent to the sequencing unit, EM lab in Conakry, 600 kilometers away, and shown to be a very close match to a virus that we had sequenced from a survivor near Zarakori 500 days previously. So the survivor was questioned, and of course he said he didn't know this woman. He did give a semen sample, which was then sequenced for Ebola virus, and we got a direct match with this young lady. And of course, with that evidence, he did admit to having a sexual relationship with her. And then through sequencing and work also with colleagues in USAMRIID, who are in Liberia, we're able to work out that the virus was spreading back over to Liberia again. We knew it wasn't another introduction by bats, and this had a really great impact on how WHO and Guinea responded. So they then they then vaccinated all those existing, especially male contacts, so that naive females couldn't be or were less likely to be infected again through sexual transmission. However, you're probably aware, some of you, that in March this year, another outbreak was identified in the same forested region in Guinea. And again, with supporting Stefan Gunter's laboratory from the BNI to do the sequencing in Guinea, it showed that the virus again wasn't a new introduction from bats, but was not that dissimilar to a person that we had sequenced back in 2015, 2016. So again, very likely to be another sexual transmission, suggesting the virus can lay dormant or persist at least for 5 years in the testes of survivors. So never underestimate what a virus can do. And you haven't got to go to far off distance lands to find interesting pathogens. This is some sequencing that we did from fecal samples from bat colonies in the United Kingdom. down in the southwest of England, in the county of Devon, and we found a virus that is not that dissimilar to SARS-CoV-2. It isn't SARS-CoV-2, even though the media obviously called it the COVID in bats. But it just indicates that there are so many pathogens around in so many different hosts out there. We just need to understand the risk of our interaction with animals. I'm certainly not proposing that we kill all bats. They have a vital role in our ecosystem. We just need to be aware of the viruses they harbor. So some of us are part of an international panel that WHO hosts yearly. And we do look at the risks of outbreaks occurring, the next pandemic. And there's a process that we use to prioritize the top pathogens for a number of parameters out there. And a lot of it is disease burden that we're seeing, which is a big gap in our knowledge. We don't do enough surveillance. But we identified these top pathogens, and the US government has a similar system. And again, those pathogens primarily are all in there, same as the UK, as well as many other countries around the world. But when we did meet in Geneva back in 2018, we did conclude that the most likely culprit for the next pandemic would be a pathogenic respiratory coronaviruses. They have all the hallmarks and make themselves a great pandemic pathogen. Finally, wrapping up, to say that we need to be prepared, and we need to work together. This illustrates the power of immunity. It's a smallpox outbreak in England back in the early 1900s. Just illustrates what protective immunity can do. And after 2015, WHO did come together to give rise to CEPI that Ricardo has mentioned already, this coalition of epidemic preparedness and innovations that has pulled together a lot of experts around the world in peacetime, developing vaccines for emerging diseases, which then can be quickly diverted to work on the pathogen that is causing the current crisis. So I would just highlight the role that WHO plays and how important international collaboration is. So I'm not going to go through these points again. Hopefully Ricardo will probably sum up at the end, but I'll just finish by saying you can never be too prepared. Thank you very much.
All right, thank you, Miles, for that interesting presentation. Thanks you for providing an overview of the threats we face and how spillover events can lead to these outbreaks. And thank you for sharing how important it is to stay ahead of these outbreaks through detection. Now, our next panelist, Erica Sapphire, will speak on effort to understand antibodies against SARS coronavirus 2. Again, that's the agent that causes COVID-19, and the global collaborations that evolved to address these questions. Thank you, Erica.
Thank you, Ricardo. I too wish I was in Texas because I am a Texan and I miss it. I haven't been there in a while. So while I'm in exile in California, we've been doing a couple of things. We've been looking at the molecular nature of how virus sees immune system. We don't know what the next virus to emerge will be, but we do know it will counter a human immune system. And so we need to understand what are the mechanisms of protection from vaccines and antibody therapeutics, and how can we steer those to be better and more effective. The other aspect of our research is to find ways to build research structures that help us as a research field untangle problems that no one lab can alone. And so this idea came right before the Ebola outbreak, when we began our first consortium in 2013, that we had antibodies that looked fantastic in test tubes, but wouldn't protect non-human primates. And we had other antibodies that would protect non-human primates, but they didn't neutralize in vitro. So there was a disconnect. We were missing some information. We were asking some of the wrong questions. And no single lab had all of the tools to figure out what we were missing. We'd also had too small a set of samples. And so we needed to build a larger pool of samples to understand the behavior of antibodies. And we needed to bring to bear more tools and more brains on the problem. And that gave birth to the Viral Immunotherapeutic Consortium. So we were asked a year ago to do something very similar for coronavirus. So for coronavirus, there were people that would have got infected because they weren't vaccinated, weren't yet vaccinated. The expectation is that there might be a mutant that would escape, or there might be a novel coronavirus. We wanted to be able to have a therapeutic in place to provide immune protection. And the challenge with antibodies, of course, is that they're more expensive and they're more difficult. You can vaccinate millions, you can deliver an antibody to hundreds to thousands. We're aiming for thousands. But to deliver an antibody to thousands and to do it cheaply enough that you can mobilize them to resource-poor regions, low and middle-income countries, you need antibodies that are exceptionally potent. You need ones that are very safe. You need ones that are remaining responsive no matter how the virus mutates or changes. And so that was our task to figure out. We launched a global collaboration to gather candidate antibody treatments from well over 70 different groups, mobilizing them from 4 different continents. Compare all of these antibodies side by side under standardized assays. And the idea is if you do the same experiment in 5 different labs, you'll probably get 5 different answers because of differences in viral passage, differences in cell line passages, difference in glycosylation in different hands. We needed to do apples to apples comparison to figure out which antibodies were best, what made them best. Are we making the right choices when we try to define what we think is best? And how to make the right combination, the right pair or maybe trio of antibodies that would hit 2 different sites on the viral spike, mitigate mutagenic escape. And how do we make this available around the world? So we're now comparing over 300 different antibody therapies that have been given to the study, and these are also the ones running through clinical trials. And they've been discovered by academics, nonprofits, small biotechs, and the major multinational corporations across 4 different continents. They've all been blinded to make the study very fair and so that we can make that data immediately publicly available. So we're building an immediate publicly available database where any researcher can look and see the characteristics of all the competing antibody therapeutics being launched against SARS-CoV-2, where they bind, how they work, if they hit a certain epitope, are they more or less potent, which kinds of antibodies against which different sites play well together. So we've gathered all these competing antibody therapeutics. We've given them all code names to make the study very fair. We've sent out identical box sets of all these antibodies to each of these experts who are looking at a different facet of antibody activity. So it could be in vivo protection in multiple different models, neutralization in different systems, structural biology, the ability to recruit immune effector functions. And so we can order them by their ability to bind to different versions of spike, different domains of spike, different mutants that are emerging in the world. We can look at the ability of each of these antibodies to neutralize in cell culture. And we're running this in 3 different labs to get a range of the different kinds of things, using either different types of assays, pseudovirus versus authentic virus, wild type virus versus the South African variant and other variants, and also get a bracket on different assay conditions. We've been able to do a lot of biochemistry, and I'm showing you data here from Dan Bettinger at Carterra and Georgia Tamaris' lab at Duke that have sorted the antibodies into bins, binding communities, the ones that bind over here versus the ones that bind over there, sorting them by competition. And so it lets us find 11 different ways that the antibodies can bind, for example, the receptor binding domain. There are at least 3 against the N-terminal domain. There are more against other ways. So earlier work in the literature defined those against the receptor binding domain, the RBD, into 4 general categories. We have so many now that we can subdivide them into 11. So if you're a Spinal Tap fan, the RBD now goes to 11. For each antibody in the database of the 300, we're going to have answers like this. This is sample number 10, COVID-10. It's right there in the database between 9 and 11. It's a human IgG1. It binds to epitope site 3A. We know its affinity for different versions of spike. We know how it neutralizes. We know its structures. And then also, because we have some beautiful tools of cryo-EM now in my lab at La Jolla Institute for Immunology, we can look at how the structure changes temporally as the antibody binds. So the coronavirus spike, the surface glycoprotein, is in red, and the antibody is in orange. You can see as the antibody binds, it twists and opens the different domains of spike. And it's also not always a complete occupancy. You don't always see 3 antibodies onto each of the monomers in the trimer. Sometimes you see 1 or 2. Sometimes they sterically occlude each other. So we can get that kind of information as well. This particular antibody has been engineered to be effectorless. Those functions are knocked out of the FC. So we can gather that kinds of information too. Now, there's a lot of data in that database, and we'll be able to use it for a lot of different purposes. And I think that's probably the most lasting effect of this consortium, is that giant body of information, lots of different levels of complexity that researchers will be able to pull out and analyze for years to come. The data I can show you today is some unpublished data using the tools of my lab, which is the structural biology and the neutralization, to see how we can attack the emerging mutations. So we've sorted antibodies into footprints and communities. And then we've solved structures. And now I'm showing you electron microscopy reconstructions of an example antibody at each of the 11 communities against the RBD, the receptor binding domain, in each of the 3 different ways that it can recognize the N-terminal domain. So the coronavirus spike trimer is in white and gray. And each different kind of antibody, and they're painted like a mohawk across the top of the spike, is in a different color. Where community 1 is exactly where the ACE2 receptor binds, and that's bright red, front and center in the receptor binding motif. Communities 2 and 3, orange, are bracketed 4 and 5 on either side, and we can look at the different sides. So to walk you through, these are the 5 different ways of recognizing the receptor binding motif. It's important to categorize these because we know which ones directly overlap with receptor, which ones are shifted to one side or the other. So when a point mutation emerges, we can know if it is or is not in the direct footprint of each one of those antibodies. We've also found other interesting things. And so this is best exemplified. If you look in the little screen, you see my hands. The receptor binding domains on spike begin in a down conformation. When the spike or the virus encounters its receptor, they lift up. Or certain antibodies that bind that same site, they lift up. And so the receptor binding domain has an outer face that's always exposed, an inner face that's only exposed in the up, and then its actual motif. And it turns out that what's exposed and what's being selected, as Miles said, for better receptor binding and mutation are these surfaces. What's hidden tends to be what is resistant to emerging mutations. So for example, the ways of binding receptor binding domain 6 and 7 are on the outer face of the RBD. 8 and 9 are also on the outer face, but they bridge a couple of monomers of spike together, so they hit a pair. They hit 2 receptor binding domains at the same time. Communities 10 and 11 are those that bind inside the spike, that inner surface, and it's only exposed when the RBD is up. It is not subject to so much mutagenic escape, and these tend to recognize the more conserved sites in spike. A lot of these antibodies that we have in the consortium against these domains have also been developed by in silico methods, and so there's something about the computational design of seeking this out that has given us candidates that we haven't always found in natural survivors. And then recognition of the N-terminal domain happens like a clock face all the way around with 3 major sites here. So I've shown you those footprints because it helps you interpret what happens when you look at binding to the different mutations that emerge. So we've analyzed these individually and together as assembled variants. And we did it both ways with the idea that the variant might change over time or something else new might emerge. So we can look at the ability of the different footprints to respond or not respond. So for example, this is community 3. This is like a lot of the ones that you've heard about in the news, antibodies being mobilized for therapeutics. They bind the top of that receptor binding motif, overlapping slightly with where ACE2 binds. And these are highly susceptible to some of the mutations like this. For example, F486L, which emerged in mink in Denmark, and K417N. Now, the antibodies that hit this footprint differ between whether that mutation lowers neutralization or completely knocks out neutralization, but they are susceptible to those sites. And so we're showing you data for the individual mutations and the assembled variants. So this kind of antibody is knocked out by the South African and the mink variants. This one, let me go back. This is community RBD6. It's one of the ones that binds the outer domain, but it's hit a good spot. It's pretty much resistant to all the different mutations except the California variant in lavender. And this is my favorite. This is community 11. This is the one that binds that interface that's only exposed when it's up. These antibodies still neutralize, almost no matter what the individual mutation is or the assembled variant. And for example, they even neutralize the Brazil isolate slightly better than others. And so this is a good one I think we should look at to try to make long-lasting antibody therapeutics. So the goals of the consortium, the reason we've been working so hard at such a tremendous level to simultaneously analyze well over 300 different antibodies and growing and 15 different assays side by side are 2. There's a translational goal and there's a fundamental research goal. For the translational research, we need to evaluate all of the competing therapeutics side by side to help the research community understand what to mobilize when clinical trial beds and dollars are limited. We've also been able to help matchmaking. So if a company has one single antibody they like a lot and they need a second one to make a pair for a cocktail, we can help them find and have helped them find complementary pairs that work well together. While we're at it, with my academic hat on, I can do something more lasting, which is building that database. So over this multi-continent, multidisciplinary study with experts in antibodies and so many different ways of analyzing and types of information and ways of dissecting antibody activity, we can come up with a landscape of activities about what antibodies do against the coronavirus, what sorts of features antibodies bring and which epitope, and which are the ones that best correlate with protection in living things. We're able to evaluate some of those assays for future use. So I'd very much like to thank my program manager for this consortium, Sharon Schindel, our program officers, Jacqueline Kircher, Tina Parker, and all of the laboratories doing all of the work of running all of those different assays. Dan Bettinger, Georgia Tamaris, Luis Martinez, Jordi Torellis, Alex McCrea, Ralph Barrick, Betty Korberg, lead alter, and so many more. I'd like to thank you for your time.
All right, thank you, Erica, for that interesting presentations. Thank you for sharing how scientists working together can create synergy and answer questions in a relatively short period of time, and also for showing the importance of understanding antibody binding to engineer better antibody-based therapeutics, especially in the context of viruses that are changing over time. Now, many of us have transitioned to COVID because of this global crisis, but what happens now? What happens after COVID? So our next panelist, Connie Schmaljohn, will present Life After COVID, how NIH labs turn to handle the pandemic. Thank you, Connie.
Hello, everybody. Thank you, Ricardo, for the very nice introduction you gave at the beginning, and also for inviting me to speak at this summit. My topic being using what we know to prepare for what we don't know. In March of 2020, our lives all changed suddenly and dramatically. At NIH, almost all research stopped, other than research on COVID-19 and some of our animal studies that we'd already started. Moreover, we were told, don't even come to work unless you're going to be doing COVID work or these critical animal studies. So in addition to all of our normal research capabilities at the IRF in Frederick, we had to develop one new capability, and that was teleworking. So now that it's about a year beyond the start of this pandemic, what have we learned during this year that would help us to prepare for the next pandemic? And I really like this cartoon that appeared in Nature a couple of weeks ago, because these women are obviously confused about how to stock the shelves for the next pandemic when we have no clue what the next pandemic is going to be. And I think this is truly a puzzling dilemma, and it is something that we need to think about in this interim. If you look at the illustration here on the left, which comes from a paper by one of my colleagues at the IRF, Jens Kuhn, you see what's termed the virus sphere. In the center is the orthovirus sphere, which contains all of the known RNA and DNA viruses, as well as all of the unknown RNA and DNA viruses. But that isn't all there is. There's also the perivirus sphere, which has things we don't know about that might or might not ever be pathogens for humans, and even more beyond that. And if we just look at where SARS-CoV-2 came from, it's one tiny spot in the RNA viruses. So clearly there's a lot more that exists in nature, and another new pathogen emergence is almost inevitable. So what do we do about that? And my contention is that we apply what we do know to what we don't know. And I think we've been doing that pretty well for SARS-CoV-2. And I believe that one of the most important things we can do is to develop animal models. We have animal models developed for numerous such pathogens as Ebola, Lassa, Nipah, MERS, and SARS, and we were able to apply these to SARS-CoV-2. They were useful and are useful testing medical countermeasures such as vaccines and therapeutics, and they can be used to correlate immune responses in pathology to what we're seeing in humans. At the IRF Frederick, we're also able to enhance imaging or enhance animal model by using specialized tools such as medical imaging and aerosol biology. Here we can use CT, PET, SPECT, MRI, or PET-CT in our BSL-4 lab, as shown on the left, and they're put into tubes where they can be observed in the cold while they are being imaged. This is just an example of a study we did with Ebola virus using a radioactive tracer to show disease progression in a single animal. So on the left, we're progressing from baseline when they were infected through day 7 of the experiment. And the yellow that you see there is uptake of fluorodeoxyglucose, which is a marker of metabolic activity, showing the disease progression as Ebola is getting worse and worse in this animal. And on the right, we're using a radio tracer component of blood, albumin, to show changes in blood volume distribution in this animal. So we were able to use these techniques to develop a macaque model for SARS-CoV-2 as well. Here we were able to instill the virus directly into their lungs, but we also used our specialized aerosolization capability to give a natural route of infection of SARS-CoV-2 to these animals, and then used medical imaging to see the disease progression. And we can not only see disease progression, we can make 3D models of what's going on in these animal lungs. And from left to right, you see a macaque with mild disease, medium disease, and then on the right, this most severe disease, which didn't peak until day 6 after infection. We also used our imaging capabilities to look at the hamster model and extend its utility. The arrows are showing interstitial pneumonia by CT in the animal lungs. Day 5 is when it peaks, day 8 is when it resolves. And again, we can use micropet scanning to look at disease progression in an individual animal. The one shown here on the right shows from baseline through day 8. This little video is a bit too long to show the whole thing, so I'm going to skip ahead. But by day 8, the animal's disease has pretty much resolved. We then can use imaging to do studies such as this, where we show that old hamsters are like old humans in that they develop a worse disease. By CT imaging here, we're seeing on the left side, young hamsters develop pneumonia that peaks at day 5, is pretty much gone by day 8. And by radioactive tracers, the same thing occurs. The hamsters pretty much develop disease by day 5, but the old hamsters continue to develop worse disease by day 8, whereas the young hamsters are able to clear the disease. So what's next? What's going to emerge from this virus sphere? I'm going to call your attention to a paper that appeared about a week ago. How accurately can we assess zoonotic risk? And predicting the next pandemic is much harder than it appears. These authors contend that we've likely identified less than 0.001% of all viruses, and that predicting which of these animal viruses might cause the next pandemic is like finding a particular grain of sand on a beach. What they suggest as an approach is that we do extensive surveillance at the animal-human interface so that we're able to detect novel viruses in humans as soon as they occur. And I agree this is a laudable approach, but it's a ton of work and it's not now being performed systematically. What we can do is what I've already talked about, apply the tools we have in better understand animal pathogens and help prepare us for the next pandemic. So I got out my crystal ball and I decided to come up with one example of something that might be next. And I chose the Arteriviridae family. This is another plus-stranded RNA virus that's taxonomically grouped with the coronaviruses. It includes simian hemorrhagic fever virus, which causes severe disease in Asian monkeys, but causes no disease in humans that we know of. In monkeys, the disease causes fever, hemorrhage, and death at 10 to 15 days. There have been up to 100% mortality in macaque colony outbreaks. And importantly, these viruses in the Arteriviridae family also infect other animals that interact with humans, such as horses, pigs, and mice. So what could we or should we be studying right now to be ready for a novel arterivirus pandemic? And again, I'll go back to animal models. We could use such models if we develop them for the known arteriviruses to study why there are these species specificities. We could look at pathological consequences in natural hosts, and we could look at what is the protective immune response in each of these species. In other words, we could apply what we know about other pathogens to understand a potential emerging viral pathogen. And with that, I'll stop and thank you for your attention.
Thank you, Connie, for sharing how COVID pandemic impacted the NIH research programs and the importance of what is known from past pathogens to future high consequence pathogens. Also, thank you for sharing how state-of-the-art technology and scientific approaches can refine animal models of disease, putting us in better position to understand pathogenesis. Now we're going to transition to our roundtable discussion. And those of you in the audience, please feel free to use the chat function or the Q&A function to ask questions to our panelists. So I'll start with a question directed to Connie since she just finished her presentation. Now, the IRF has a lot of state-of-the-art equipment. How do scientists in the audience partner with IRF if we have some project of interest?
Thank you. That's a really great question, and I would direct you to our website, the NIAID IRF website. But basically, how we work is all through collaborations. We are not an investigator-driven research organization. We do some research, which we call our toolbox projects, to build capabilities. But other than that, we work exclusively with collaborators, such as Erica, who's been a wonderful collaborator. And how it works is the collaborator will propose to one of our scientists a project they'd like to conduct. We bring it to our internal committee to talk about, see if it fits within the NIH mission. And if it's an NIH funded project, that's all that needs to happen. If it's an NIH grant or other NIH funded. If it's not funded by NIH, we have one more level of scrutiny by a scientific steering committee. But once that's passed, then we actually pay for the entire study, other than the purchase of animals. There's no charges for labor, per diem, any of that kind of thing. So we're a great deal. So any of you in the audience that have good ideas of things you'd like to partner with us, we'd be happy to hear them.
Great. Thank you, Connie. That is a great deal. Now, the next question is, based on your experience and research, what are the next pathogens that may potentially start a pandemic? I know, Connie, you hit on this a bit. So why don't we start with Miles and then go through the panelists and see what your opinion is on this question. Miles?
Thank you. Yeah, I mean, I referred to the meetings that are held by WHO that try to pull together all the disease burden data on the things we do know about, because they do cause outbreaks. And obviously Ebola has been known since 1976. But what they're most likely to be are caused by unknown relatives of the things we do know about. For example, SARS-CoV-2. We knew about SARS-1 and MERS-CoV, but obviously one that we don't know about that's even better at transmitting. There's an advantage over the viruses that are out there at present. I mean, it all depends if it's an outbreak or pandemic. A lot of the ones we already know about will continue to cause outbreaks until we've got effective vaccines and decent diagnostics all over the world. But surveillance is key to helping us understand what's coming next.
Great, thank you, Miles. Erica, would you care to comment?
Paramix of viruses, a combination of two things. Some of them, like measles, have an R naught as high as those we know, 16, 18. And others have lethality, like Nipah and Hendra, up to 60, 70, 90%. That's a bad combination. And they infect humans and they infect animals. So there could be a novel paramyxovirus or one that we already know about.
Great, thank you. And Connie, you wish to expand a little bit?
Oh, I'll just go along with what Miles said on the pathogen X work. After a lot of discussions at these meetings, we decided that it's impossible to prepare for an unknown unknown. So what you do is prepare for an unknown known, basically, a relative. And I think Miles explained that already, but that's, I think, all we can do.
And the one thing that they all have in common is no matter what the pathogen is, it's going to encounter a human immune system. Even if we went to Mars and encountered a novel Martian pathogen, our human immune system is going to have to figure out how to counter it. And so development of the toolboxes of vaccines and treatments and understanding how to bolster lifelong immunity is going to be key.
All right, great. Thank you. Erica, what do you think we can gain from a large collaborative effort versus efforts that occur in individual labs or individual research programs?
You very much need both. So an individual research program has the advantage of speed, right? And so if you need to come up with a therapeutic fast, or you want to get to a paper fast, a single group or two group, small group going alone will get you there faster. And we need that speed, we need that innovation. You need the larger collaborations when the question is more complicated than any one group has the tools or the samples to address it alone. So if there is some black box that we haven't yet untangled, so for example, our black box in Ebola, we completely understood how mechanical neutralization happened, how the antibody anchoring to the spike could block receptor binding, block conformational change, and that biochemistry and that biophysics of how an antibody would block a viral entry in cell culture, we understood that. We had the structures, we had the samples that would do that. What we didn't understand is how that antibody would inspire immune control of infected cells or immune clearance of the virus from the body. And that turns out to be really important too. Nobody had those tools because it's complicated. It's a lot of transient interactions. It's different in every species. It's hard to model. And so that's a greater effort to build a bigger body of knowledge for the future. And so to understand that, to tease out the signal that you hadn't found before among the noise, you need more antibodies. You can't start with the 20 or 100 your own lab raised. You need 1,000 to down select to the ones that look at, the more interesting features. And you need lots of different tools. If you don't know what experiment is going to give you the signal you're looking for to find them, you might need to analyze 12 and look at a really large data set to figure out what's going to be the important one. So you need both. You need competition to drive the speed and the innovation. You need collaboration to answer greater questions and build new tools.
Great. Connie, do you have thoughts on that as well?
Well, obviously I want the collaborations because that's what we do. But I always have found that you get a lot more out of other people's ideas feeding into your own rather than trying to keep everything tight to the vest and making your name for yourself without including the best science that you can get through talking and interactions with others.
Great. And Miles, I think at PHE, you probably have a very similar experience.
Yeah, I mean, our model is not that dissimilar to Connie's, but we do ask you to pay for the experiment or co-fund it or get a grant together. We're not so flush with the cash that Connie is, but it's a lovely position to be in and a really, really clever way of working. It's a great idea of NIH. So yeah, we have our own investigator-led programs, but because we have the BSL-4 facilities for the UK government, we obviously collaborate with all the academics who don't have access facilities, and we do all the non-human primate research, infectious disease here as well. So, that's been my life for leading this department for the last 12 years, is that we collaborate not just with other academics. It's important to collaborate extensively with the commercial sector because they're the ones who bring drugs to market. And again, from this outbreak, we've worked with many, many vaccine developers and manufacturers, as well as those developing therapeutics. So collaboration, of course, is a no-brainer. And I think we missed some of the after speaker bar sessions to do extra talking about how we could work together. And I think I'm really looking forward to getting back to that. Though virtual meetings like this have done a really good job in the present climate.
Great, thank you, Miles. Now, I did get a text here from somebody asking about Ebola virus, since you mentioned Ebola virus. They're asking that previous outbreaks were relatively limited. What contributed to the large outbreaks we've been seeing lately with the Ebola viruses?
Well, I mean, so the DRC outbreaks, have been quite large. Obviously, many factors contributed to the West Africa outbreak, which were primarily due to the response being so late. And by the time it did happen, the virus already spread through all the main networks from Gekadu, the epicenter, which straddles really Sierra Leone and Liberia, into those capital cities, having very poor infrastructure for public health, obviously was an additional factor. And I mean, I don't believe the virus mutated to become more transmissible. Some people say that. I just think it happened for the virus to be in the right place at the right time.
Thank you. Here's actually a follow-up from the same person texting me. So Erica, with regard to antibody therapies, Do you think that's the way to go with Ebola viruses and other filoviruses?
I think for people that haven't been vaccinated and you have an emerging outbreak, I think it's an idea. For someone who's been newly affected and there's that time where the viral load is ramping up, they definitely work really well. And it's better than the alternative, which is hoping for the best. To draw rings around people and to cover thousands to millions, a vaccine is the best bang for your public health buck. But those antibodies will treat the people that are already infected.
Great, thank you. Okay, so here's one. So we are all part of WHO ad hoc COVID working groups. And have the WHO working groups or similar working groups contributed sharing information and real-time data? And how has the collaboration impacted science? And Miles, I'd ask you to first respond to that before the other panelists.
Yeah, so my personal experience with WHO has been extremely positive. They're not the most perfect organization in the world, but there isn't a perfect organization, in my opinion. But from the outset, back in February, where some of us attended the first WHO-sponsored COVID R&D roadmap meeting, there was between the talks, we had better sharing of information, and then the subsequent working groups, especially animal models, is one that I was most involved with. I definitely shared data before it was published in preprint, which changed. Somebody was in one of the groups who was about to go and do a challenge study in ferrets. We just told him exactly what we found, and they modified their protocol. So that's a really good example of the impact it can have. So for me, they've been extremely valuable.
Yeah, I think that's an important point, Miles, that you brought about altering your protocol. So that part of it we've noticed is the limited resources, especially when it comes to non-human primates. And I think overall, sharing these information also helps us to refine our studies and use minimal amounts of animals. Erica or Connie, can you share from your perspective on the WHO committees?
Well, I'm not going to talk about the committees. Well, I guess they're committees per se, but I think that the WHO has made a huge impact in equitable distribution of virus throughout the world with their COVAX facility, which is an organization that they've set up along with Gavi and the Gates Foundation and some other funders to find a way to get vaccines all over the world to countries that don't have their own ability to do that. And they've partnered with UNICEF. And I think that is a real important part of the WHO response to this pandemic. Also, I think by integrating a lot of their work with CEPI's work, that it has expanded their capability to see what's going on and to respond to it in a more timely fashion.
I agree. Infectious disease is a global problem. It needs a global solution. We have to pull up all the chairs to the table.
Great, thank you. So here's a question, Connie, on hantavirus. So you're the chair of the hantavirus group, and the question is, how does the outbreak we had back in the 90s compare? I mean, of course, the COVID pandemic is much more robust, but what are your thoughts with regard to the differences between that outbreak and the current pandemic?
Well, thank you. Bringing up my very favorite viruses, of course, although we don't work on them here at this facility I'm at now. But the big difference is that the hantaviruses were not person-to-person transmissible. So they may have been more deadly with 40% mortality, those ones that appeared in the US, but it was very rare that you would see human transmission. It was only one particular, none in the US. Andes virus in Chile has been found to be person-to-person transmissible. But if that were something that happened, here you go with another pandemic.
Erica, here's one in the chat. Let me pull it up real quick. Your thoughts on structural studies on how dimeric IgA or IgM bind to spike protein? Would these isotypes be better in neutralizing SARS coronavirus 2?
That's a great question. And the answer is I don't think we know, because I think they've gotten insufficient attention. I think we have focused primarily on IgG for the affinity. But I think that avidity and that protection of the mucosal surface are key, especially early in infection. I think that's something we need to do. I think those molecules were also out of reach from our earlier tools of X-ray crystallography, but now it's something that we can do, and it's something that I'd be very interested in doing.
Great, thank you. How do you think, Erica, having the structural data has influenced our ability to better respond to these pathogens, these new emerging pathogens?
Well, the immediate response of taking care of patients and controlling that's not helped by structural biology. Structural biology is helped on the figuring out what is the right thing to mobilize and what kind of protection are we getting at. Can we predict using structural biology that this will be a lasting antibody, or will it be escaped or emerge? We can also now, using structural biology, look at a polyclonal antibody response. So we can look at what's the range of antibodies of people living in this village that may have been exposed to things over the years, or the range of things elicited by vaccines. So it's very much a complementary tool, but it's going to be a couple of steps behind Miles's group.
Great, thank you. So Connie spoke about the challenges that IRF faced with doing research in the middle of a pandemic. Miles, would you care to comment on what kind of challenges you had there at the PHE?
Yeah, no. So our facility is specifically maintained for emergency response. So it was 100% of the scientists in the lab, because they were all redirected to COVID-related duties, from testing lateral flow systems to growing up virus for animal models, et cetera, et cetera. Obviously, all the non-laboratory staff were asked to stay at home, but all the lab staff were here working, obviously through the night, which probably was pretty stressful. And getting access to materials was difficult, because everybody wanted all the PCR reagents, which wasn't helpful. Getting access to animals for us was a lot easier, because we have secure supplies of non-human primates. We have a couple of colonies here in Porton Down that we maintain to ensure supply, specifically in situations like this. But we did find people in the universities who obviously shared their equipment with us and reagents, so it was pretty collaborative. But obviously the big drive was all about diagnostics in the early times, and trying to retool all the academic groups and industry to provide that massive diagnostic push.
Oh, great. Thank you, Miles. Erica, can you comment on your facility?
Yeah, we are cryo-EM, protein engineering, and antibody discovery. So we have 2 of the 300 kiloelectron volt electron microscopes. And these can get us structure in hours to a couple of days instead of previously needing years. So when I say we're a couple of steps behind Miles, I mean, we can get an answer of what are we looking at, and what are the surfaces we're looking at, and what do the antibodies react, while he's still there. So it's not the years behind that it used to be. It's more like 2 days behind. And the other instrumentation that we have is the ability to detect at the single B cell level what each antibody is doing. So we could take a survivor or a vaccinee's B cells and line them up and pen them one by one and interrogate each individual B cell they make and say, of the different antibodies you make, which one reacts to Ebola, Sudan, Marburg, and Bundibugyo? That's the one we want. We lift it out, boom, that's the one we're looking for. Or you can just profile on a broad level. Which of these antibodies cross-react between Lassa and Lujo? Which of these antibodies are specific? Which of them recognize the pre-fusion trimeric viral surface version? Which ones recognize the sprung less useful versions? We can get that precision level of interrogation of what is the structure, what is the right antibody recognition, what is the wrong antibody recognition, and we can now do that from the polyclonal sera.
Great, no, thank you for that. If I can just share a little bit about Texas Biomed and how the pandemic impacted us. So we have a range of pathogens that we study, including the BSO4 pathogens. And for us, it was much like Connie expressed and you guys expressed, in which we had to mobilize our staff to work on COVID-19. So essentially, those individuals focused on SARS coronavirus 2 were the ones here in site. We were fortunate to receive some funding from the community to do some philanthropic work on developing models. Also, BARDA helped us to contribute to those studies as well. So we were essentially needing to respond to this threat. We limited people on site, and that also helped us to be able to maintain the health of the individuals working with the animals as well. So now we have a question here. Oops, I just lost it. 1 second. Oh yes, it's about the variants. So this is for the general group. Do you think the variants that are emerging now will be an issue with the current vaccines?
I'm happy to go first. From the analysis that I've seen and been involved with, I don't believe we have a variant yet that is going to cause major problems. There's probably one around the corner, but I believe that at present, the vaccines will do an adequate job. They will at least keep people out of hospital with severe disease and death. So I don't think we've seen the scary variant yet, but not to say there isn't one on the horizon.
Is the rate that we're seeing variants something that could have been predicted? Is this virus replicating or mutating at a higher rate, or this is something that is common and is predictable?
Well, RNA viruses mutate. That's just what they do. They don't have, many of them have no proofreading capacity. Coronavirus has some. And so it doesn't mutate as quickly as those kinds of viruses like Ebola or HIV that have none. But what we're seeing here is that because it has infected millions of naive people and occasionally has infected immunocompromised people where their immune system failed to get control, so there's constant mutation, constant sampling, it was inevitable that these kinds of things would emerge.
Now we do have a question here. If you had a magic wand, and this is for everybody, that you could wave and fix a major challenge in your field, what would it be?
Connie, it's your turn to start with that one.
Well, I already got my crystal ball out. Now I have to get my magic wand out too. So I think that probably what I would try to fix is in my field is, I'm saying this very broadly, in my field, I think what needs to be fixed is equitable distribution of resources throughout the world. So my field I'm taking as biomedical sciences and human health care, and the ability for underprivileged areas of the world to have the access to the resources that are needed for healthcare. I think we would avoid a lot of these things like Ebola outbreaks if what Miles said was to happen, that the resources weren't so constrained, that the response was quicker, that the people had adequate protective gear. I know that's not really my little molecular virology world, but that's what my magic wand would fix.
Erica, what are you thinking?
I wish we had the knowledge base to be able to predict what the right vaccine platform is. It's still very much empirical. Do we display something on a viral vector? Do we make mRNA? Do we make protein subunits? And it seems to be largely empirical about understanding what is going to create a lifelong response, like a one and done, like the polio vaccine, and what you have to boost and will achieve breakthrough with. I think that we need that better understanding of how to recruit long-term memory for an entire population.
So if I could fix anything, obviously I'd choose what Connie had just said, which is obviously so important, more important than anything that we do individually. But one fix might be for emerging diseases would be a decent surveillance system where we were joined up internationally and we did invest in capability in all countries to sample the viruses that we think we know are out there, but get more information on that, and that will help us prepare as well, and it will help us shut down outbreaks more rapidly, which will obviously save lives as well.
Oh, thank you. So leading on to that, there's another question here about surveillance. Have the variants caused any issue with the current surveillance technologies? Have these variants escaped any of our detection methods that are currently out there?
Yeah, I mean, if I can go first. So a lot of these variants were picked up by diagnostic labs because they knew some of their primers were working and binding and some weren't. So that gave an indication either the diagnostics were failing or the virus was changing. And then around the world, we're sequencing a lot more. We've been sequencing under a thing called COG in the UK very early on. And for concentration of virus, we've probably got more sequences than any other country in the world. And that's the way to work it out quickly. And we've also set up now with WHO backing an international sequencing capability with hubs around the world, again, working in unison with WHO.
Great. There's a question about SARS coronavirus 1 here in sequencing. So it looks like it took several months to get the sequence of that virus. Now it sounds like it's much less than that. How has understanding the sequences led to advances against the current virus?
I'll give that one to Connie, SARS-1.
Well, you know, those were different times, and that was more of an unknown. I mean, SARS-2 was like SARS-1. So I think that helped a lot. But the sequencing has been absolutely crucial for everything from making the vaccines, making the diagnostics, understanding the immune response. So the advances in sequence technology that were made between when SARS-1 was first around and SARS-2 have, you know, been leaps and bounds. And I think it's probably the most important thing that has helped with the fast response to this virus and this disease.
Great. So before I start wrapping up the session, would you like to provide any final thoughts?
When there's a new virus, it does take a little bit of time to go and figure out what platform is going to work and develop a new vaccine and develop a new series of diagnostics. We had not been in the habit as a nation of running so many diagnostics. You go to the doctor, he just figures out if it's bacteria or virus. If it's a virus, we used to just be sent home and said, drink plenty of fluids, get some rest. So this is a fundamental shift and a fundamental expectation that we want to be able to know what it is that people are infected with and have ready and expensive tools to do it.
My final thought is I'd like to give a shout out to CEPI. We are so fortunate that organization came into existence at the time it did, that had prepared laboratories, funded vaccine development efforts on known and unknown pathogens to be ready for the unknown, to include MERS, SARS, you know, things that weren't necessarily going to be big pandemics, but they were thought of as examples of things that could have a virus like them become a pandemic. So I think we'd be months behind if CEPI hadn't already been in existence and funding some of these efforts.
So I would just say, especially ditto on the CEPI front, we've worked with them for several years now, and they've been phenomenal on making things happen. Because without funding and without resource sharing, which they've helped immensely, you can't do a tremendous amount. I think in peacetime, we should be trying to find this magic antiviral. I know that a lot of defense organizations, and our own one in the UK, want you to come up with this magic drug that can kill all viruses. But maybe we should put a little bit more effort in peacetime, at least trying to find a panel of antivirals that can be utilized when we're hit with an unknown. So preparation in peacetime.
That's great. Now, Erica, Connie, and Miles, thank you so much for being with us today. I appreciate it. The Institute appreciates your participation, and the audience, I'm sure, appreciates it. And we're grateful for your time and your continued efforts against this pandemic. And we hope and we look forward to your work against the next pandemic. I also like to thank our colleagues and future colleagues and friends in the Institute that are listening to this first Global Health Symposium. And today we learned that the virus fear is enormous and we have only scratched the surface. Thus, it's incredibly hard to predict the next viral pandemic. We also understand that surveillance and serial epidemiology would be really helpful to predict and identify new outbreaks of pathogens and the risks they pose. We learned that we can apply existing tools that we have, such as in the IRF, imaging modalities, animal model development to study disease courses of known pathogens and advance our understanding of future emerging pathogens. And we do know that a better understanding of pathogenesis of known pathogens will help us prepare for the next outbreak. Finally, we learned the power of collaboration, the ability to have international multidisciplinary approaches to untangle some problems that no one group can handle alone. And we understand that collaboration can coexist within the framework built on competition, and they can proceed side by side on complementary studies. Well, I've enjoyed this session. I hope you have as much as I have, and we're looking forward to seeing you all at the second Global Health Symposium in the future. And for the audience attending now, you'll be able to attend another meeting in about 10 minutes, so you can log on to those currently. Thank you again, panelists, and thank you for attending.
Thank you, Ricardo. Bye-bye.
Thank you.
Thank you. Good to see you.